Okay.
Last two sections.
Woo!
Dave, are you with me?
I'm with you.
All right. So the last two sections are. I don't want to spend too much time on it because I want to have you work in these magnets. But basically, what this section is saying is that you don't just have to have glucose. You don't just have to break down glucose to make ATP. There's other stuff you can break down to make ATP. As a matter of fact, any of the organic molecules we studied, specifically proteins, fats, and carbohydrates, those can all be broken down at some point. Now, let me grab them over here. Do y'all know, do y'all remember our organic molecules? So, what is this one I'm putting together here? That's a protein, a chain of amino acids, right? And these, of course, are carbohydrates, like glucose. And what's that thing?
Fatty acid.
It's a fatty acid chain that's part of a fat. So these can all be utilized by the mitochondrion to make energy, to make ATP, to harness the energy to make ATP. So this is just showing you, the whole process. There's glucose that you start with. The glucose is converted to pyruvate. So what process is this going from here to there? That's glycolysis. And then the pyruvate to acetyl-CoA, that's the prep reaction right there. You see that? In this big circle here, what do you think that is? Citric acid cycle. So this is just kind of showing how glucose gets broken down. And notice there's also some backwards arrows. The glucose can be broken down to this phosphoenol pyruvate before it forms pyruvate. That's part of the glycolysis process. But it can also go backwards. This phosphoenol pyruvate can be put together to form glucose. So you'll see some backwards arrows here. All of these words here, do y'all recognize any of those? Where have you seen those before?
Are they types of amino acids?
Bam! Cartier.
Yeah.
Fantastic. These are all amino acids. So these amino acids can tie into the process. You can have your amino acids. I can't read it. My eyes are too bad. Methionine. Do you see methionine up there anywhere? There it is. Methionine. This amino acid right here could get in the mitochondrion and attach to or become part of the citric acid cycle and be broken down further. It ties in right here at this point in the citric acid cycle. Now, I didn't have you have to memorize all of these things, but basically what happens, do you remember the structure of an amino acid? There's a carbon in the middle. What else is there on an amino acid? There's an R group. There's an amino group. There's a carboxyl group, an acid group. Good. There's an amino acid. In order to take an amino acid and break it down and tear it apart to make ATP, you have to remove this NH2 group. It's a process called deamination. The NH2 group is taken off and it becomes a waste. And it actually gets in your blood, and your liver will pick it up and will put a couple of these together to make a molecule called urea. Have you ever heard of urea before? That's the yellow stuff in your pee. So, whenever there's yellow stuff in your pee, that comes from your body has been breaking down proteins for energy. Taking the amino acids, taking proteins that maybe you ate, I don't know if you ate steak or something, you take the proteins, you break them down, you can burn that for energy.
What's the process called again?
Deamination is the removal of the amino group. And you see the rest is just carbons. And these carbons are the same as the carbons here. So as you put this, as you attach this, remember you break this apart, and these carbons go in here and attach right here. Remember, 2 carbons attach right here in the citric acid cycle? Mm-hmm. Y'all remember that? Mm-hmm. Okay, well, the 2 carbons could have come from pyruvate, but they could also come from these amino acids. You see what I'm saying? So they don't have to necessarily come from glucose. Also, here is a fatty acid chain. You see how it's got a whole bunch of carbons on it? Basically, these fatty acids can be broken down for ATP. There are enzymes that will break apart the fatty acid and leave just sets of 2 carbons. And then those 2 carbons can feed into the citric acid cycle, just like these 2 carbons did, and be broken down. So your fatty acid chains can be broken down too for energy. So you can see fatty acids enter the system right here. Actually, the fatty acids, let's say we have an enzyme that will break that fatty acid chain off. Here's 2 carbons. That can come in the mitochondrion, and bam, attached to the CoA, and now you have 2 carbons. There's also some hydrogens attached to it, but that can feed in right here to the oxaloacetate, just like it was a piece of glucose. So you can make ATP using amino acids and fatty acids as well as carbohydrates. Do y'all know the molecule glycerol? Mm-hmm. Where did you see glycerol?
It's that
What phospholipid?
The top.
Well, actually, it wasn't there. It isn't a phospholipid.
It's like, I assume it's the top part of the phospholipid.
There was glycerol. I have magnets for this, but I don't have them out. Glycerol attached to 3 fatty acid chains. Y'all remember that? Made of fat. Mm-hmm. So that glycerol, that can be torn off the fat and can be used, take part in the glycolysis reaction. The fatty acid chains can be broken down and be part of the pyruvate oxidation. A bunch of amino acids, actually there's amino acids that can be broken down in glycolysis, amino acids that can be part of the pyruvate oxidation, and amino acids that can go directly to the citric acid cycle. So amino acids can contribute anywhere here. So the basic idea of this section is that your body can use any of the organic molecules for energy. And it's not just your body. This doesn't just happen in people. All life. It have plants, fungi, can use molecules at any portion to get ATP. Now, the last section talks about how the process of breaking down molecules is regulated. I just want to show you one example. Look at how insulin works. Have you ever heard of insulin? People who are diabetic, they don't make insulin. And watch how insulin reacts. Watch how insulin causes the cell to be able to absorb glucose molecules. So here's an insulin molecule. It attaches to a cell. This is a cell. The insulin molecule attaches to an insulin receptor. There's a chain reaction that happens inside the cell, and we have a whole chapter that talks about these chain reactions that happen inside a cell. But once the insulin is received, there's a chain reaction inside the cell that causes, there are actually vesicles sitting inside your cells that have a channel protein called GLUT4. GLUT4 is a channel protein that's attached to the membrane that the vesicle is made of. And it's just sitting inside your cell. So when the cell receives insulin, this vesicle fuses with the membrane. Do y'all remember exocytosis, when the vesicles fuse with the membrane? Mm-hmm. Well, that's what this thing does. And so when insulin is received, this vesicle fuses with the membrane, and now you have this GLUT4 receptor on your membrane. And you can see the purple there? That was originally the membrane of the vesicle. And now you see it's part of the cell's membrane. And now we have the GLUT4 channel right there in the membrane, and now glucose will come into the cell. And that glucose can be broken down for energy. So the cell can control, or the glucose could be stored. Either way, the cell can control how much glucose goes in it with some sort of molecule like insulin. And that's what we're talking about. So when we say that these processes have some control, there are ways to control the process. This is control over the respiration process. If you don't have glucose in your cell, you can't break it down for energy. If you need to break it down for energy, some insulin could be released from somewhere, could attach to the cell. Now the glucose gets in. Now you can break it down for energy. Or store it. A lot of cells will just store it, like your liver cells will store the insulin. Not break it down for energy, but just store it for later use. Isn't that a neat way of controlling glucose? Here's one more way that this process is controlled. All these reactions here, this is glycolysis. We start with glucose, we go through a whole bunch of reactions, 10 of them, before we end up with pyruvate. I just show it to you by breaking the molecule in half. But you actually have to go through steps to do it. And there are control areas along the way. Like, for instance, there's an enzyme that does this first step. It converts glucose to glucose 6-phosphate. We can stop that enzyme by having an inhibitor. Remember when we studied enzyme inhibitors? You had allosteric inhibitors and you had competitive inhibitors. Well, we can inhibit that enzyme. That's one way it's stopped. Then the glucose will not be broken down. It may diffuse out of the cell. That's a good point of regulation. Here's another point. This part right here is where glycolysis is usually inhibited. There's an enzyme there that I can't read. It has a real big name. But anyway, we can inhibit it there, and that stops the process. We have inhibitors that work down here on this enzyme that stop the process. So the cells can inhibit the process. Sometimes you need to break down glucose, and sometimes you don't. So there are inhibitors there that can inhibit those enzymes. Inhibition of enzymes is another way where we can speed up or slow down these processes. All right, we got about 10 minutes left. I need a few more people to do these, and we'll just keep doing them until everyone's done them. Thank you.